Hi everyone, welcome to this week's discussion on transcriptional regulation. Today we'll be exploring a number of topics such as what transcription factors are, how they work, and how transcription is regulated in prokaryotes, which includes bacteria and archaea, and eukaryotes, which include plants, animals, and many other types of organisms. Before we begin our discussion on what transcription factors are, though, we should talk about what transcription is in the first place. Transcription is one step in the process called the central dogma, in which DNA, the instructions for all of life, is used to produce a functional product, such as a protein. Transcription is the step where the DNA in genes is transcribed into mRNA, also known as messenger RNA. mRNA is then used in a separate process known as translation to encode a specific order of amino acids to produce specific proteins. Transcription factors are proteins that regulate this process, so in other words, they control how much or how little transcription occurs for a given gene. All transcription factors have a DNA binding domain. Four common structural motifs in these DNA binding domains are the helix-turn helix, the helix-loop helix, the zinc finger, and the leucine zipper. Each of these motifs has a separate mechanism that allows its efficient binding to the DNA. A helix-turn helix motif, for example, contains two helices which sit in the major groove of the DNA. A helix-loop helix motif and the leucine zipper motifs both promote the dimerization of transcription factor proteins. A zinc finger motif contains a zinc ion which helps stabilize the motif and hold it together, allowing it to bind to the DNA. Ultimately, what really matters about different transcription factors is the role that they play in regulation. Some transcription factors, known as activators, bind to regions of DNA adjacent to genes called enhancers to cause what is the upregulation of a gene. Others, such as repressors, bind to similar such sites and cause the downregulation of genes. Now that we know what transcription factors are and how they work, let's take a look at operons, which are a specific type of transcriptional regulation mechanism that exists only in prokaryotes. To understand how these work, let's take a look at a schematic of a well-known repressible operon known as the trip operon.
In bacteria, the trip operon is responsible for synthesizing tryptophan when it can't be taken in from its surroundings. The trip operon consists of a regulatory gene, promoter, operator, and three other genes which encode for 5 different enzymes. The regulatory gene codes for an inactive protein known as a repressor. When tryptophan is present, it will act as a co-repressor and bind to this repressor protein. The repressor will then be activated and combined to the operator region of the operon. This will block RNA polymerase from transcribing all 5 genes which encode enzymes that synthesize tryptophan. On the other hand, when tryptophan is absent, nothing will happen to the repressor. As a result, the repressor will not bind to the operator and transcription will continue smoothly. The 5 enzymes will then synthesize tryptophan. Another operon in bacteria, known as the lac operon, is responsible for using lactose as an energy source. This operon essentially works in the reverse of the trip operon. In the lac operon, there's a regulatory gene which codes for an active protein known as a repressor, as well as a promoter, an operator, and 3 proteins which can help it use lactose. When lactose is present, an analog of lactose, known as allolactose, will bind to the active repressor protein and render it inactive. In this case, allolactose can be described as an inducer. Contrary to the trip operon, this prevents the repressor from binding to the operator, and the 3 genes which help use lactose are transcribed. This makes sense because when lactose is present, it's beneficial for the bacteria to have the ability to use it. Similarly, when lactose is scarce, allolactose will not be synthesized in great amounts, and the repressor will remain active, bind to the operator, and inhibit transcription of the 3 genes. So, what did we cover today? Well, we began by talking about what transcription factors are and how they function to regulate gene expression. We also talked a little bit about their protein structures, and then went on to describe prokaryotic gene regulation with 2 key examples, the lac operon, which is a type of inducible operon, and the trip operon, which is a type of repressible operon. Thank you for watching, and we hope you learned something new today.